Good afternoon, ladies and gentlemen, and distinguished guests of honor. It gives me great pleasure to introduce Professor James Noble of Columbia University in New York City as the keynote speaker of the 5th annual Gabby Williams Alzheimer's Foundation Lecture. Dr. Noble is a recognized leader of the American Academy of Neurology. He is an expert in dementia, behavioral neurology, and concussion, and was the director of the Medical Student Neurology Clerkship for many years. Dr. Noble is also a renowned scholar of the distinguished Virginia Apgar Academy at Columbia University. But perhaps what is most special for me today is that Dr. Noble has been a friend and colleague of mine over the last 2 decades, in which we have struggled and we have triumphed on the front lines of academic neurology. And so it gives me great pleasure that Dr. Noble is the 5th annual speaker of a lecture dedicated to my father, Dr. Gabby Williams. And so without further ado, it is my pleasure and honor to introduce to you Professor James Noble of Columbia University. Thank you.
Hello, everyone. I'd like to thank the organizers of this conference for inviting me to speak. It is my pleasure and honor to be a part of the Gabby Williams Alzheimer's Foundation 5th annual meeting, not only because of what Gabby Williams means to so many of us, but what Dr. Williams, his son, means to me. Dr. Williams and I have been close colleagues in neurology here at Columbia University Medical Center for close to 20 years. My name is Dr. James Noble. I'm an associate professor of neurology here. I specialize in Alzheimer's disease and am the clinical core leader and co-director of our Alzheimer's Disease Research Center funded by the NIH. Today, I'll be speaking about what's new in dementia, from biomarkers to treatments and research, what's happening now and what's coming soon. I hope to share with you some of the excitement and promise that we have in the field, but also better understand what challenges we face to ultimately find effective treatments for a group of very unfortunate diseases. My disclosures are here. The most relevant ones are that I draw much of my support from the National Institutes of Health, and I'm the volunteer president of the nonprofit Arts and Minds, which has the web address there, and I may be reviewing briefly some off-label medications for dementia care. Today, I plan to present to you the progress in dementia that has been made over the last 25 years, roughly spanning my entire medical training and clinical career. It's been an exciting time, but also one that gives us a pause to say, what can we learn in the next 25? I'll be speaking about the basic dementia definitions, how we originally framed these around clinical definitions, are moving towards a biological definition and maybe merging those back with the clinical definitions as well. And talking about how we've made these diagnoses over time and how we may make them in the future. I'll be speaking about the epidemiology, or the frequency with which these diseases occur in both Africa as well as the rest of the world. And I'll be focusing on symptom-based therapies, as well as new treatments and others that are in development, and what directions may be ahead of us in order to make some inroads towards better treatment. Let's start with why this matters everywhere. Obviously, I'm not there with you, but I know that this is a serious issue for the many people that are there at the conference and many beyond. As of 2014, there were more than 1 billion people in sub-Saharan Africa, representing about 1/5 or 1/6 of the world's population, and that population is growing. Not just in general, but with its increasing life expectancy. And a report just last month highlighted that there has been a 10-year increase such that over the last 20 years, such that children born today are expected to have a life expectancy of around 63 to 65 years. And that is nearly the world's average at this point. And by 2030, the implications are that more than 67 million persons will be aged at least 60 years and above in sub-Saharan Africa, the ages at which dementia begins to become more prevalent. We know that this matters also because it's a complex disease. There are uncertain pathobiological pathways towards its development and also uncertain treatments. It is nonetheless an intense area of study globally, and we need to make sure that works for many will work, what works for some will work for all. Early diagnosis, we know, is the key. And Dr. Williams and I have been working for years to try to better understand how can we minimize the challenges that many face in establishing a diagnosis, whether it's something neurologic, whether it be dementia or stroke, trying to establish a diagnosis at an earlier time. There are unique challenges faced in every locale, from cultures to language to traditions, and this impacts our understanding currently of how commonly it occurs in various places throughout the world. We also understand that the current diagnostic pathway is not entirely practical in most places, and we'll talk about how we may be able to reconcile that sooner rather than later. And we also recognize that long-term care needs, caring for the caregivers, are especially intense and take many hours away from other obligations. It becomes costly to families and also psychologically and physically demanding on many. And you can see here at the right is a figure from the International Journal of Alzheimer's Disease in 2014, highlighting the fact that right now we need to better understand its epidemiology, the epidemiology of dementia throughout sub-Saharan Africa, so we can better understand how to make those treatments more effectively delivered. And there are inroads happening right now all around us. But the key questions I often think about as I take care of my patients with dementia, and the questions they often ask me that I'm going to try to answer for you today, include establishing the differences between the terms dementia, mild cognitive impairment, also known as MCI, and normal forgetfulness that all of us experience throughout our lifetime. We'll also talk about the most common causes of dementia, how we make a diagnosis of dementia, and some new research findings from the field that help to highlight all of these things. We'll talk about the American framework for national and local research infrastructure and help understand efforts that are underway in Nigeria and the surrounding countries to better understand and establish a local and national research infrastructure throughout Africa. We are getting closer to a blood test for Alzheimer's disease, and we'll talk about what that may look like and what the implications may be. But ultimately, we need to understand, when will we have treatment? So let's talk about how we, for many years as physicians, have been trying to translate complex information into a diagnosis. At the left are three individuals who are central to the history of Alzheimer's disease. At the top left, you see Dr. Alzheimer himself, a German neuroscientist who first met and described this woman here, August D., back in the early 1900s. He established her diagnosis clinically. She was a woman in her 50s who was developing forgetfulness, and eventually she died, and those are his hand drawings of the microscopic changes that are still known as Alzheimer's disease changes. Below him is Solomon Carter Fuller, who was a Liberian neurologist, or Liberian-born neurologist, who eventually became the first African-American neurologist in America, and was known to have worked on and studied under Dr. Alzheimer and brought many of his lessons to the States. And still, what those physicians did in the early 1900s, it's largely the approach that we still take today, and that is acquiring a patient's story. We understand their problems, we ask them questions, they tell us the problems they've been experiencing, their family members share their concerns, and then as physicians, we generate a hypothesis, we establish a diagnosis, all based on the background of what knowledge we have, the context in which these concerns arise, and our experience in establishing a diagnosis effectively. But more and more, we're shifting towards better understanding how to make a diagnosis using what we call biomarkers. And these are objective tools like brain scans, sometimes blood tests or spinal fluid analyses, to help us better understand how the brain ages, or how it ages differently from a normal population, and how we can identify when there may be something of concern like Alzheimer's disease. We have more advanced technology available to us, including PET scans, which can highlight the changes that happen in a brain down to the molecular level. But the greatest challenge of all is, especially in establishing a treatment, it's all fine if we have all of these diagnostic tools, but what does that say if we cannot establish a treatment? Also, we recognize that these diagnostic tools are not available everywhere. So what can we do to not only understand how to make a diagnosis effectively, but make an accurate one, even when we may not have the resources that are conventionally used? Well, let's start with defining the term dementia. So dementia is a disorder of cognition or behavior that largely works in or largely begins to manifest as one of the main cognitive functions, or the overall functions of the brain, begin to change. You can see here at the right a cartoon description of how the interlocking functions of the brain occur. And most obviously, we think about memory loss in Alzheimer's disease and dementia, but certainly we think about the language function, visuospatial abilities, processing speed, and executive skills. These are the abilities for us to think about one idea to the next, juggle many things at once, plan ahead. All of these things we need intact in order to get through our normal day-to-day routine. In dementia, we start to see that there's a change in at least one of those domains. It usually begins with a single one, maybe several all at once, or one maybe most apparent. And in the case of Alzheimer's disease, it's nearly always memory, but sometimes other findings that do change at first. With all cases of dementia, there's a progressive involvement of multiple domains, such that language abilities are lost, or maybe executive abilities, or something like visuospatial function, or even slowing down, processing speed declines. There's a background called cognitive reserve, and that's basically the sum of all experiences that people have had across their lifetime, ranging from professional to educational experiences. These may play an influencing role in how changes within the brain are manifest in day-to-day life. And we also know that dementia is nearly always acquired, or that's really part of the definition, and it's almost always over the course of aging, but somebody doesn't necessarily have to be age-ed. We rarely will see patients with Alzheimer's disease and related dementias as young as the 30s and 40s, but most of the time we're thinking about patients who are presenting in their 50s and older, and most often in their 70s and older. And dementia is also defined by having a meaningful impact in day-to-day life. Many of us experience problems with our thinking and memory, but it's really a critical threshold that begins to define dementia, not just that there's been a change, but a meaningful change that ultimately requires some assistance from somebody else in order to get through the day. We can also identify what we call localizable features. We can probably, at least based on clinical history, point to an area of the brain or maybe several areas of the brain that aren't working as they should. And we now also know that this is not just about these kind of five broad, more cartoonish looking domains I'm talking about, but each of the functions of the brain can really map to these intricate, very small regions of the brain, so we know exactly where memory lapses begin to occur. We also know where language changes can occur and frontal executive problems too. So what is the difference? We've talked about dementia. How do we define that relative to something called mild cognitive impairment, or what's normal forgetfulness? Cognitive aging, MCI, and dementia occur on a spectrum, and with the one end of the spectrum of normal cognitive aging, we recognize there are a range of changes of thinking and memory considered to be normal for age. And generally, there's a mild deterioration in something like processing speed and memory, and that's often the case across many individuals. We may forget where we've placed something or forget what we plan to do, but it usually comes back. And most often, those kind of benign things that we see are not an immediate precursor to something more. And we know what is considered to be normal based upon normative data across languages, ages, and cultures, thousands of people having taken the tests. Mild cognitive impairment is something a little bit more. We see that these are changes in thinking and memory that are not just considered to be normal for age alone, and is often a precursor for dementia. And without help, someone nonetheless remains independent in all regards. And this stands in contrast to dementia, which we just defined, in which there are clear needs such that somebody's reliant upon somebody else in order to maintain their independence. They need assistance with their medications, getting to appointments, remembering their calendar. And then that's often the case in the earlier stages of disease. And later on, somebody may need assistance in the basic activities of daily living. That is, they need assistance with going to the bathroom, getting themselves dressed, maybe preparing a meal, these kind of things. What are the most common causes of dementia? Well, we think about dementia as kind of an umbrella term, and beneath it are a whole host of other neurological problems, or more specific neurological problems. We can think about them in a group of diseases that we call neurodegenerative disorders. It's a mouthful, I recognize. But the general grouping of these, we'll get to it in just a minute, is very intentional. These are all these three common diseases that we see in dementia, called Alzheimer's disease, Lewy body dementia, or dementia with Lewy bodies, and frontotemporal dementia. They all share in common a gradual progressive course, such that there is a clumping of different changes within the brain, and eventually leading to the clinical symptoms that we see in day-to-day life. We also recognize that vascular dementia, or stroke-related dementia, is exceptionally common, especially in areas that have very high frequencies of things like high blood pressure, cholesterol, diabetes, and stroke itself. Coronary disease is another risk factor associated with vascular dementia. In contrast to the degenerative disorders, vascular dementia often occurs either in a stepwise or even sudden manner, such that an area of the brain is injured by a stroke, leaving somebody reliant upon somebody else because of those areas of thinking and memory that were ordinarily affected in things like Alzheimer's disease. Gradually over time. In contrast, in vascular dementia, it happens suddenly. At least in most studies, we recognize that there's actually rarely the case of one change, but actually a multitude of changes that we see under the microscope. That is, we see Lewy bodies with Alzheimer's disease or Alzheimer's with vascular changes. And at least in many studies that we've looked at of the brains, including thousands of brains, we know that the older one gets, the more likely one is to have several pathologies. And when somebody has several pathologies under the microscope, they're more likely to have clinical symptoms of things like Alzheimer's disease. But also we want to recognize that there are many other causes and ones that are potentially treatable and reversible. Depression and schizophrenia, mental health disease. We also recognize that there are medical conditions such as a low thyroid or B12 deficiency, a vitamin deficiency, or common infections like HIV and neurosyphilis. We also will do brain scans specifically to try to make sure we haven't missed something structural like a brain tumor or too much fluid within the brain. And rarely we see rapidly progressive diseases that are associated with inflammation or autoimmune conditions. And you may have read to some extent in the press about certain athletes having a condition called CTE, but at least as of now, that condition is thought to be fairly rare. So what do all of these neurodegenerative disorders have in common? Well, they have a common pathological conditions in which the neurons lose their function, their structure, or both, leading to the neuronal death. Neurons or nerve cells, these are the key brain cells that really are required for all of us to function well. When they fire well, our brains work well, we're able to think, walk, talk, and so forth. But when they begin to progressively become injured, such as in neurodegenerative diseases, they eventually become sick and get to shrink and wither and even die. Even though the brain is still there, these individual cells are getting sicker over time. So together we call these neurodegenerative disorders. This is a large group of heterogeneous disorders, probably even numbering in the hundreds, even though we have single names like Alzheimer's, Lewy body disease, frontotemporal dementia. The closer we look, the more we realize that there are probably many specific subtypes that we just don't have the capacity to specifically name just yet. I think that's probably where we'll be in some time. They're also typically adult onset conditions, and they progress in a relentless manner for unknown reasons. And the singular thing that we see under the microscope is this clumping of abnormal protein, what we call aggregates of these depositions, both outside the cell and inside the cell. And what's become most fascinating in the last few years is, although we are clearly very good at identifying these changes under the microscope, what is now not known is whether or not if one takes these changes out of the brain, that is, if somebody could snap their fingers and come up with a new medication that took those proteins and those aggregates out of the brain, does it actually make a difference? This is a critical thing that we're trying to work on right now, and it's not actually as clear as we thought. So aside from those singular cells getting sick, what we recognize also is that there are certain areas or parts of the brain that are especially vulnerable, the selective vulnerability of a population of nerve cells. In the case of Alzheimer's disease, it happens to be the nerve cells that are involved in memory circuits. Again, they all have this gradually progressive nature of these aggregation, these misfolded proteins. We also know that it's not so much necessarily specific to the protein, but where that change happens in the brain is the type of clinical problem that we see. For instance, if there's a microscopic change in memory circuits, we'll see memory loss. If there's a problem in the front part of the brain, somebody may have executive or behavioral changes. And sometimes we even see these changes in the spinal cord, leading to things like amyotrophic lateral sclerosis, or ALS, a progressive weakening of the muscles. And again, we have these lingering questions about these pathological changes. Do they actually cause a clinical syndrome? Are they a marker? Are they a bystander? Does it make a difference if we take it out? And if we take it out and there's no difference, does that mean it was too late? Or have we hit the wrong target? Are we way off the mark? As I mentioned previously, the last 25 years have been especially promising in both dementia discoveries and progress, despite the fact that we don't have especially good therapies just yet. When I started in my clinical training in the late 90s and into the early 2000s, by that time we recognized that brain atrophy and shrinkage and functional patterns could be established on brain imaging. We had modestly effective memory medications, which have become available, and they're still available today and remain a central part of our treatment. They work relatively briefly and in a relatively modest effect, and we'll talk about that in a moment too. But it was also the dawn of time when we can start to identify Alzheimer's disease in spinal fluid, the same fluid that's in our back is in our heads, and it can be drawn out in a safe manner, analyzed especially, and we can identify with a high degree of confidence whether or not somebody has Alzheimer's disease causing thinking and memory problems at present. It was also the early days of when we started using a brain scan in order to highlight the changes of Alzheimer's disease using a brain scan called an amyloid PET scan. In 2003, between then and 2021, we had no new drugs that were approved for Alzheimer's disease, but we started making progress in how we began to make a diagnosis. And by 2020, we had promising blood biomarkers for Alzheimer's disease. And we also had a new PET scan highlighting the other main pathology that we see in Alzheimer's disease. In 2021, the new medication of a new kind called aducanumab came along, and we'll talk about those challenges it presented in just a moment. How do we diagnose Alzheimer's disease? We talked about this dementia umbrella term, Alzheimer's disease being the most common one, and it's probably the one that we're mostly familiar with, either through our own family experiences or talking about it with others. But all persons who have Alzheimer's disease have a gradually progressive, insidious onset. They also have a clear indication that this is a change. This is not something that they had since childhood, but has become acquired. And we begin to define the kind of Alzheimer's disease that is a forgetting kind, which is amnestic, or a non-amnestic or non-forgetting kind, such that we talk about the first symptoms that began with the most prominent ones that were apparent over time. There may be a language presentation or visuospatial or executive problem that emerges. And whatever is the central symptom at first is usually the form that we call it, even though we recognize that the changes microscopically are largely the same. Again, why these changes occur in certain areas of the brain in some people and other areas of the brain in others, we really don't understand. But we recognize also that about half or more of all persons with dementia are thought to have the primary cognitive changes due to Alzheimer's disease. But again, when we look at these series of autopsied brains, it's actually exceptionally rare to find pure Alzheimer's disease. And then one of the most fascinating things I learned a long time ago that really challenges all of us is that about 30% of people who die will have Alzheimer's changes identified microscopically, and they had no detectable changes, even through very rigorous testing shortly before they passed. So what does it mean to have Alzheimer's disease changes? Is it something that's bound to begin to affect somebody clinically? Or is it a bystander? And again, it challenges our notions even of how to diagnose somebody, especially when it comes to screening. When it comes to the epidemiology, most of what we know about certain risk factors come from a series of US studies, which have identified that there are at least 6 million Americans, most of whom are over the age of 65, who have Alzheimer's disease. We now recognize it's between the 7th and 8th most common cause of death. And there was a large jump in that number just a few years ago, only because it became reconsidered when it came to autopsy reporting or death reporting. We also know that, at least in the United States, the lifetime risk of developing Alzheimer's disease once somebody hits age 65 is about 15%. And that's because the life expectancy is into the late 70s or 80s for many individuals. And age alone is the greatest risk factor that's been recognized in Alzheimer's disease. Where I work here in Washington Heights, which is in northern Manhattan of New York City, we've come to recognize that there are differences in one group to another, especially when it comes to race ethnicities. But we've also come to realize that the race ethnicity is really a crude proxy for complex lived experiences, including educational heterogeneity, language and acculturation, health and income inequalities, structural racism, and all of these things have very complex interactions with one another, as well as genetics. But we have to start somewhere in identifying there are differences, and now we have to uncover why these differences are here. And some of my colleagues have been especially committed to this, and has led to major discoveries, including understanding that some genes are relative to some individuals and not relative to others. Some risk factors are relative to some individuals and some not to others. This is foundationalist understanding the risk factors for Alzheimer's disease in our community and are informative in other centers too. I want to highlight somebody named Hugh Hendry, who is now retired, but started what became really the first African study of the epidemiology of Alzheimer's disease. Back in 1991, he envisioned developing a project based in Indianapolis, Indiana, as well as in Yoruba, to identify were there differences in the dementia risk for individual factors in different contexts between African-American families living in Indiana and African families living in Yoruba specifically. And eventually they followed about 2,500 individuals from each site. Perhaps somebody here in the audience had a family member who was within that study, made that commitment. And the goal overall was to understand what factors may have influenced the development of dementia in both groups. There were complex assessments of demographic, lifestyle factors, and genetics, understanding that there were probably some differences in either vascular or environmental risk factors, and then people were tracked over time through clinical and neuropsychological data. Key findings were clearly that just like every other place, we see that age is probably the most important factor in dementia expression. There were some findings in some of the earlier studies suggesting that the rates of dementia were lower in Nigeria, but after adjusting for age in some of the subsequent cohorts, when the cohorts were allowed to age sufficiently, there actually were very few differences between the African and the African American communities. There were, however, some differences in things that led to complex interactions between environmental risk factors like diet and genetic factors as well. And they highlight what we've seen in nearly every study the world around, that it is important to understand these interactions in order to determine dementia risk across various populations. I want to make sure we spend a few moments talking about dementia with Lewy bodies and frontotemporal dementia. Dementia with Lewy bodies, at least in many studies, has suggested to be the second most common cause of dementia behind Alzheimer's disease. In the United States, there are about 1 million people who are estimated to have Lewy body dementia, compared with about 6 million with Alzheimer's disease. Alzheimer's disease tends to affect slightly more women than men, but in dementia with Lewy bodies, it tends to be far more male, with 4-to-1 ratio of male versus female. We see many cognitive features overlap with Alzheimer's disease, including forgetfulness, executive problems, and visuospatial difficulties, but it has unique features, including what we call a fluctuating cognition, whereby there may be good days and bad days that are distinctly in contrast to one another. There are recurrent visual hallucinations, which are well-formed and detailed, and there's a really profound degree of dream enactment, people acting out their dreams, talking in their sleep, maybe even having things like fighting in their sleep or acting out jobs that they used to have. There's also what we call Parkinsonism, stiffness, slowness, tremor. It's not Parkinson's disease, which is maybe for another time, but we do see Parkinson's changes under the microscope, but in different areas of the brain that we see in Parkinson's disease. It's not the major feature, but it is a common feature. In frontotemporal dementia, it tends to be one that we see very often in younger individuals under the age of 65, where it's tied with Alzheimer's disease as being the most common cause of dementia. Over the age of 65, it becomes far less common. We see cognitive decline with some overlap with Alzheimer's disease, executive problems and forgetfulness, and Parkinsonism there too, with unique features including personality change, lack of motivation and disinhibition. Some individuals have profound language changes with loss of information and loss of fluency, the ability to speak clearly and quickly. And about 10% may have developed amyotrophic lateral sclerosis, which is a weakness based upon changes in the spinal cord. It also has a strong genetic component in which about 1 in 5 will have genetic change, but only about 1 in 3 will have a known family history. And although we haven't talked about Alzheimer's disease and its genetics, we know that there are about 5 dozen different genes that are associated with Alzheimer's disease expression, making it slightly more or slightly less common. And there are 3 different genes that are exceptionally rare, but are known to run in families who have Alzheimer's disease beginning when it begins in less than age 60, and especially if multiple generations are affected. Vascular cognitive disorders, such as MCI and vascular dementia, are really follow stroke. We know that, at least in the United States, stroke is the 5th leading cause of death. It's the 3rd most common cause of dementia. And what's really fascinating is about 3% of all people have a stroke in life, and about 30% of autopsied brains will have a stroke, suggesting that symptomatic stroke is far less common than asymptomatic stroke. There are 3 main causes of stroke that we see: from large vessel disease, where we have blockages of the large vessels. We see small vessel disease, where there may be blockages or even bursts of those small vessels that cause strokes over time. And we also see things that where it's not so much blockages of the vessels due to changes in like high blood pressure, but actually a specific change associated with blood vessels, almost unique to Alzheimer's disease persons, where the same Alzheimer changes that are happening in the brain substance happen within the blood vessels too. They lead to hemorrhagic strokes throughout the brain. And depending on where these strokes occur, if it's one or if it's several, we may see different manifestations of dementia. I know this is a lot on this table, but this is also what we're trying to assimilate here, trying to understand, okay, well, we have 1 brain change in Alzheimer's, or 2 brain changes in Alzheimer's disease. They share features in amyloid angiopathy. There's change that we see in Alzheimer's, also in frontotemporal dementia. Why do we see all these common findings? We don't yet know, but it's interesting, all of these linkages, and we're trying to find common factors, common genetic factors to better understand these diseases. So where we're moving now is that the diagnosis of dementia continues to evolve, and it follows a lot of the research findings from the field. Right now, as I said at the beginning, Dr. Alzheimer's and I continue to diagnose patients phenomenologically. We take the history, we do an examination, and we start with the key question: is this person forgetting, or does it seem like something else is happening? We try to identify reversible causes. I gave you a list before. One I should also mention is that sleep is especially problematic for people with memory problems, and we often see that sleep apnea can cause a severe sleep deficit and may be associated with problems with thinking and memory, not just in the short term, but in the long term. And that's one thing we also put on our list. We also want to try to identify in our minds what's the most likely underlying cause, the microscopic change that helps us inform a prognosis and treatment plan. And where we are in the future, maybe even now, is that we're really trying to move towards that biological characterization to track and identify a problem as early as possible in hopes of identifying disease-modifying treatment strategies also as early as possible. So right now, in most practices, including a primary care doctor's office, a brief cognitive screening test is essential for helping to identify a cognitive problem. But it's also the case that we use that in conjunction with family reports, people around somebody, to indicate whether or not they think there's a problem. We recognize that about half of all people who have Alzheimer's disease have no awareness that they do have any significant memory problem. After that, we move towards screening for reversible causes, making sure we haven't missed something like a metabolic problem, a vitamin deficiency, or rarely infections that aren't otherwise recognized. And a brain scan usually can help us exclude possible things we don't pick up on examination, although that's quite rare, things like a tumor, a stroke, but it also increasingly helps us identify what areas of the brain are affected and changed in somebody who has dementia. We've moved towards this new framework that we talk about called ATN, and that stands for amyloid, tau, and neurodegeneration. And basically, this has become the central piece in how we think about diagnosing somebody in a research framework based upon biological indicators, but we're also thinking about how we apply this to the clinical practice, and it's already in place in some centers. Again, we're thinking about how do we transition from a research framework to better diagnosing and delivering therapeutics, recognizing, however, that pathology precedes cognitive changes. And we recognize that Alzheimer's changes can begin as early as 10 to 20 years before the very first symptoms. We also conceptualize neurodegeneration like cancer, thinking about, is there a transformation time where it goes from an earlier simple tumor into something that's much more malignant and rapidly progressive, which we see in many of our patients. It also helps us begin to recognize the combinations, these dozens of combinations of clinical features, yet we only see a handful of these primary and identifiable pathologies. And it also helps us understand about notions of pathobiology of disease and the changes of the disease through the biomarkers. As I mentioned a moment ago, there's the A, the T, and the N. From a practical standpoint, we can now begin to identify this A part of it through spinal fluid analysis and brain scans, but the practical aspects of those are very challenging. Many centers don't have spinal fluid analysis capabilities or the personnel to be able to handle a large load of individuals. And so the promise that we see now is that there do seem to be several different blood biomarkers, several which are becoming approved in the FDA system towards helping us to identify several of these biomarkers. They're not necessarily quite yet ready for prime time, but if these really begin to hold the promise that we think they do, it would be a real life-changing experience if we can begin to diagnose Alzheimer's disease accurately and at the time that we need based upon a blood test alone. But there are problems with biomarkers. So the challenges, again, availability, especially of imaging, and this notion that I mentioned a moment ago, that about 30% of persons over the age of 65 die with microscopic Alzheimer's changes in the brain but have no symptoms. And biomarkers have to be used cautiously. The promise is, as I mentioned, if it's reliable, is early detection, early treatment, and hopefully a broad rollout. We recognize that there are nationalized research models, but they always begin locally. And at our center, we have more than 30 years of research experience in direct partnership with the community, listening to our community, serving them, helping them out, and working with them to better understand how cognitive aging occurs. And we have more than 12,000 local community members who've been involved in aging research over this time. We use, as I mentioned, community advisors, as well as other stakeholders in our community. We have multiple ongoing studies focusing on Alzheimer's disease, normal cognitive aging. Some have brain imaging, blood, and spinal fluid studies, and all offer direct studies of the brain microscopically after the time of death. Overall, we've had about 800 brains that have been donated for the purposes of research over the last 20 years. And basically, our approach is, if somebody's interested in getting involved in research, we want to find a study about brain aging that's right for them. In the US, there is a nationwide network that's been put together over the last 20 years called the National Alzheimer's Coordinating Center, which itself is based in Seattle at the University of Washington, but helps to harmonize and collect data from about 40 centers nationwide. Collectively, this is I realize, a busy figure here, but it's all to say This is what it takes in order for this to occur effectively, where we have sample collection, we have data analyses, we have data storage, and we have sample storage. And collectively, we now have over 40,000 individuals who've participated across these different studies, with genetic data available on many, many more in related studies too. Encouragingly, just earlier this year, the Africa Dementia Consortium published in the journal Alzheimer's and Dementia, and they showed several key things. And this map at the right is from that article, which I encourage you to take a look at. And it shows very effectively where there are centers of excellence, which is the head that you see there. These are both organizations in supportive care, but also potential research and academic institutions that are focused on things like Alzheimer's disease and stroke-related dementia. You can see the DNA molecule, which is interestingly showing where there are known to be early onset or genetic causes of Alzheimer's disease. And while this is the case that we think that there are so-called foundry mutations in certain areas of the world, I will just say that the more we look, the more we find that there's hardly a place now that doesn't have some sort of unique family history causing a unique neurological syndrome. It may just be that these are the ones that are now known. And finally, in the different colors, you see the degree with which there are different genetic studies and epidemiologic studies taking place. New frontiers in biomarkers, as I've said, I think within the next few years, there will be much more availability of amyloid biomarkers. There are now 2 that are becoming available in the United States. Not yet ready, not yet available are these other biomarkers called tau biomarkers. And we also have new PET scans that are kind of on the horizon. But again, the rollout and the pragmatic issues around PET scans are problematic. However, we need to do these where we make sure that we link the clinical spectrum to the spinal fluid, to the PET scan, to the blood work, in order for us to assure ourselves that the blood work in the end is as effective as the more complicated studies. And what we all want to know, and I'll finish up with this, is, what are the effective Alzheimer treatments that we think about? This is a graphic I often turn to think about, okay, well, where are we on these different curves? The nihilist in us might think that we're on this yellow curve where we have no effective therapies or anything that we offer is as good or no better than placebo. But in truth, for the last 20 years, we've been working with this red curve where all of the medications that we offer, things like, and I'll show you a list of medications in a moment, but things like donepezil and other related medications offer a brief symptomatic benefit, but ultimately do not impact the long-term care needs or hospitalization, institutionalization, or even death of our patients with Alzheimer's disease and our family members. However, we are hopeful that we will begin to move towards these blue and green curves where we even stop or slow the progression of Alzheimer's disease. There's an additional curve not on this graphic, and that is recovery. And we'll hopefully get to that at some point too. Dementia treatment options are several now. We recognize that there are symptomatic medications focused on memory. We also have several medications, symptomatic medications, focused on behavioral problems that are especially problematic with the middle and late stages of disease. But we also have non-pharmacological therapies, knowing that hearing aids, exercise, diet, and managing comorbidities like stroke are especially effective in the earliest of stages in potentially warding off or maybe slowing down. Some of these are yet to be proven effectively, but they seem to hold a lot of promise, at least epidemiologically. And finally, on this end of the spectrum are new biological medications. These are especially complex in order to deliver. This one in particular is a monthly infusion requiring serial MRI assessments and is associated with a fair degree of complications, making its practical use very, very challenging. But it may be that this is the first of others to come and provides us a paradigm for thinking about new medications down the line. All of these medications, no matter how you slice it, end up being what we call palliative therapies, where really the goal is to relieve suffering of dementia caused by cognitive and behavioral decline. This is not to say this is end-of-life care, hospice care. This is palliative, which is the definition by the World Health Organization used here. Aducanumab is this monoclonal antibody, which I've talked about briefly already. It was fairly controversial when it came to be approved in the United States. It went through an accelerated approval with a lot of controversy around whether or not it should be accepted. It ultimately was, and was found to be quite costly, anywhere from $28,000 to $50,000 a year, plus the infusion administrative costs, plus serial MRIs that have to be done over time. Its uptake has been slow, but again, it may serve as a with other medications to come. I want to show you a bit of hope here with this target graphic that comes out every year from the Alzheimer's Drug Development Pipeline working group, showing that there are many different drugs that are in the development, from the earliest stage of phase 1 to the closest to the finish line, phase 3. You can see that this is a very crowded field, and if you looked at the ones from 10 years ago, it would be quite empty. We also see here that the mechanism of action is much longer. The list was maybe 2 or 3 points long, and now it's about 10. It's also the case that we have people that are being treated in all stages of disease, looking down here from healthy volunteers to the earliest of symptoms to even those who don't yet have any symptoms but have biological indicators. This is what I see on a regular basis, and I think something here is bound to work, and it gives me hope for the future. Let's not forget that this is not about just the biology of disease, but about caregiving itself. Dementia caregiving is costly, especially reflecting on the lost income related to informal caregiving, and can have a long-lasting psychological distress and toll on family caregivers. There are effective caregiver programs that offer the patient and caregivers simultaneously, if at all possible, and the goals of care are to provide activities and new opportunities, engage in recent or nascent interests, and improve apathy and depression. And for the caregiver to help them see a community of support through shared experiences, and hopefully to improve depression and stress, self-efficacy, and general health. In New York, we've been offering programs both in person before the pandemic and virtual ever since through a nonprofit that I started called Arts and Minds. This is a part of really a global effort to try to help through non-pharmacological strategies by spending something like an hour or two at an art museum on a weekly basis. And we have at least preliminary findings to suggest that it really connects with certain family members and participants, both patients and caregivers alike, in improving their spirits. Whether or not this has a role long-term remains to be seen, but there's enough promise that I'm very actively involved with this. And our programs themselves have been offered more than 400 times in the pandemic and more than 1,000 times in person before it. We're very excited to continue to offer these for free. And if you'd like to learn more about Arts and Minds and possibly how something like this might be delivered in your community, please contact me afterwards. The way I see the future, and I hope this is going to be part of my career, is that there will gradually be a continued embrace of complex brain changes and pathways recognizing that they create similar clinical phenotypes of very different diseases. We recognize right now that we need to have minimally invasive, cost-effective therapies, maybe even transitioning just to blood work, and maybe even getting to a point of just using these tools for screening rather than diagnosis, hopefully being able to clinically and biologically diagnose somebody effectively if possible. While you may have heard terms like precision medicine, the mathematician in me must remind you that this is actually something that we don't really aspire to, with the target here at the top right being precise, where all the answers are together, but they're off the target. What we want is accurate medicine and really patient-centered tools over time. So hopefully these multimodal biological treatments will become available to us, and we'll continue to offer patient-centered precision medicine or personalized therapies really in order to make people's lives better. I know there's been some talk about this book called Navigating Life with Dementia, and I'm exceptionally proud to say that I spent close to 7 years writing this book. It just came out, and there are several copies being made available, I know, at this conference. And if you're interested in getting one, hopefully you'll get one there, but there are others elsewhere. I don't feel that comfortable pushing a book, but this is to say, this is a passion project of mine, and one that I hope somebody here in the audience draws some benefit from in better understanding this disease. Here are the key points that we cover, including all the things I've covered today. How do we diagnose dementia? How do we understand its progression? What are the most common causes? What are the treatments of cognitive impairment and the behavioral and psychological symptoms of dementia? I go into everything we've talked about in much more detail, especially the notions of how do we help to support somebody through the later stages of illness? How do we monitor and maintain independence, safety, mobility, and advanced care planning? And with that, I want to thank you. I want to thank Dr. Williams and his family once again for inviting me to give this talk. And I look forward to hopefully meeting some of you at some point. And as always, if you have any questions or comments for me, feel free to reach out, james.noble@columbia.edu. And if you happen to be one of the people with the book, please let me know what you think of it. Thank you.